Good morning, everyone. My name is Marlies, and I work as research project manager on this project on single-cell profiling of different cancer types before and after immune therapy. And so to start, I'll just briefly show you what immune therapy and in particular immune checkpoint inhibitors are. So in tumors, tumor cells can interact with T cells, and this interaction switches the T cells off, so the tumor cells are not recognized by the immune system. And so immune checkpoint inhibitors can block this interaction, and this way the T cells can be reactivated to recognize the tumor cells and to kill off the tumor. And so one example of immune checkpoint molecules or an interaction is the interaction between PD-1 on T cells and PD-L1 on tumor cells. And this interaction can then be blocked by specific antibodies recognizing PD-1 or PD-L1, and these are then reactivating the immune response. And so these immune checkpoint inhibitors have been recently developed, and they are, in the last few years, being approved for a number of cancer types. The first one was melanoma, but in the last year, many other cancer types have followed. But quite a big problem with these immune checkpoint inhibitors is that the response rates are highly variable among different cancer types. And so in some cancers they work really well, in others it seems that they're absolutely not working. And therefore it's important to determine biomarkers that will predict response to these immune checkpoint inhibitors. And so therefore we set up a study across different cancer types, and this is the design of our study. So we receive pre-treatment biopsies from cancer patients. Then these patients get one cycle of immune checkpoint inhibitors, and then a second biopsy is taken or the tumor is resected and we receive a sample. And so on these samples we perform single-cell profiling using multiple techniques in order to get more insight into the mechanism of action of these immune checkpoint inhibitors and also to ultimately identify a biomarker for response. And so this whole project is being carried out in Leuven at the KU Leuven and the VIB Center for Cancer Biology. Just to show you, we are collaborating very well with the university hospital, but also with industry. We have a team, the VIB TechWatch team, who scout for new technologies and help us implement them. There is a whole platform on disease modeling, and also there are multiple groups working on bioinformatics and developing new methods to actually analyze all these new data. And so just to show you, our center is really suited for this kind of project because the university hospital is literally next to the research building. So the patients come here, and here is our center. So the tumor samples can get from the clinic to the research lab in less than 30 minutes. And so these are all the cancer types we are currently profiling. So there are already many included, and we're still actively looking for collaborations with the clinic to extend this. And so also one big aim of this project is to share the data. We're not generating all these single-cell data just for ourselves, but we really want to share the results of our efforts. And this will be done through the launching of calls. And the first call will be on the breast cancer samples. We already have 30 matched before and on treatment breast cancer samples, single-cell sequencing data. And so this will be launched soon, so you can all apply to see our data. And so the breast cancer trial is one of the most advanced ones, and we have collected the most samples from this, and that's why I can already show you some preliminary data from this. So our first analysis included 13 breast cancer patients, and we performed single-cell RNA sequencing and single-cell T-cell receptor sequencing. And you see here a plot showing more than 80,000 cells, and we can nicely identify multiple subtypes which are found in these tumors. And so when we zoom in on the T cells, we can recluster them, and we can identify additional subtypes of T cells in which we are mostly interested in the CD4 and CD8 exhausted T cells, since these are the T cells that are being reactivated by the immune checkpoint inhibitors. And as you can see, indeed, if we look at PD-1 expression, this is mostly found in the CD4 and CD8 exhausted T cells, the populations which we are interested in. So then we tried to classify the 13 patients we had into potential responders and non-responders. And we did this based on the T cells we found in the tumor. So first group of patients we think are potential responders because we find a high number of T cells in these patients and also a high number of exhausted T cells. Then the second group of patients presents with a high number of T cells, but there are almost no exhausted T cells. So the other T cell subtypes are present, but the exhausted T cells do not seem to be present in these patients. And then a third group of patients are also non-responders because they just have no T cells at all. They're very cold tumors. And so indeed, if we look at PD-1 expression on the T cells in these different groups of patients, we see that indeed in the first group, from which we think are potential responders, they show high PD-1 expression. While in the second group and the third group, the non-responders, we don't see this expression. And also, if we look at T cell expansion, we can depict this on the T plots by coloring the dots of the T cells that are actually expanding. We did this using T cell receptor sequencing. We can see that in the responders, it looks like the T cells are actively expanding, while in the non-responders, we don't see this effect. So nothing is really happening in the T cells. And so I only had 7 minutes, which is a very short time. There's much more analysis already being done, so you can come to my poster and we can discuss. Also, many of my colleagues have posters in this event, so please be welcome to also have a look at their posters. And I need to thank lots of people who work on this project, and thank you for your attention.
Thank you very much. Do we have questions?
Hi. There's this great, fairly recent paper on the role of ADAR in this context. Did you look at ADAR1 expression?
No, not yet, but that's a very good suggestion. We will definitely do that.
Hi, great presentation. So in the breast cancer data where you're showing the preliminary results, so you have TCR data before and after immunotherapy?
Yeah.
And so can you comment on the clonal distributions before and after in the matched samples? Are they in the patients that have T cells? Are the same clones present after immunotherapy? Do you see changes in expansion?
We see that the same clones are present, but in a higher frequency. And we also see new clones emerging after immunotherapy.